OCRAT Biology Revision Experiments Investigating Photosynthesis by SHS Science. This video is going to focus on two experiments. The first one is the use of a photosynthometer and the water weed Elodea, and the second experiment is where you take extracts from a cotyledonous plant and make discs from them, and then look at the rate of the discs going from the bottom of a beaker or syringe to the top. The first experiment discussed is the use of a photosynthometer in the measuring of the rate of photosynthesis. Generally, light intensity is investigated where you'd have a lamp at varying distances to investigate how quickly the Elodea in the diagram you can see photosynthesizes. Key details you can see in the diagram are the fact that the Elodea is in the test tube and that there is a capillary tube with a small funnel above it. The idea is that the Elodea will produce bubbles of oxygen, and those oxygen bubbles will be collected in the capillary tube, and the length of the oxygen bubbles will be measured, and the total volume of oxygen produced over a certain period of time will be able to be measured. For this experiment, there are some key details that need to be discussed for the experiment to work reliably and accurately. Firstly, the Elodea, it needs to be cut at an angle to allow the oxygen bubbles to actually be produced. Ideally, this is actually cut underwater, and it's kept in the same type of water that it is has been left in. This will ensure that the changing in water mineral content or any of things like that won't have an issue with regards to rate of photosynthesis. Also, the joinings between the syringes, the plastic tubing, the capillary tube all need to be airtight, ideally using something like Vaseline petroleum jelly to make sure they are completely airtight so that there is no air loss and possible effects on photosynthesis. And finally, and probably most importantly, is the fact that the plant needs to be allowed time to equilibrate to the conditions. So generally, once the plant is being cut and set up, you need to allow between 5 to 10 minutes for the plant to get used to its conditions, so you feel confident that the rate of the oxygen that is being produced, the rate of photosynthesis that's being measured, is an accurate and fair one. If you're investigating how light intensity affects photosynthesis, the key variables that you need to control: firstly, wavelength. Only use a white source of light. If you were to change the source of light, such as using a slightly red or yellow bulb, that can obviously change the wavelength of light and therefore whether the plant absorbs the light or not, and therefore actively uses that light for photosynthesis. Temperature of the water. Ideally, you'd use a heat shield in between the light source and the plant to ensure that any heat from the light is absorbed. Carbon dioxide levels are controlled by the amount of sodium hydrocarbonate, either powder or solution, that is put into the water. This provides the plant with carbon dioxide that is dissolved in the liquid. Obviously, it's quite an easy way to control this, as it's just simply the amount of sodium hydrocarbonate put in that is controlled. And finally, the plant. You need to use the same species of plant, and ideally the same plant throughout the experiment. Elodea is the classic example. The reason you need to try and keep the same plant is because different plants in that species will photosynthesize at different rates, and therefore you want to try and ensure that the plant you use is the same throughout, so that you are more sure that the results you get are the results you should be getting. The idea of this experiment is very simple. It's to do with the density of the leaf discs. The leaf discs will decrease in density when they photosynthesize by the fact that the plant will produce the carbon dioxide when light is shone on it, therefore causing the discs to go from a sunken state at the bottom of the beaker to rise to the top. In order for them to sink to the bottom, all the air needs to be taken out of the spaces between the spongy mesophyll. This is done by using a syringe, as you can see, with a finger being placed on the top of the syringe and the syringe plunger being pulled out, removing all the air. Those leaf discs can then be put into the beaker, and the experiment can be done. This experiment can be done with light and shade plants, or it can actually be done with any form of plant, ideally that are cotyledonous plants.